According to FDA, validation is a method to establish documented evidence which provides a high degree of assurance that a specific process will consistently produce a product meeting its predetermined specification and quality attributes. Validation of all major pharmaceutical systems is essential. It ensures the quality of manufactured products and helps to manufacture the quality products. Need of validation. It helps to investigate the deviations in manufacturing process. The risk of regulatory non-compliance is minimized after validation. Increases production due to minimized rework and rejection. Reduces the production cost of product. Batch to batch variation is minimized. These are the validation parameters. According to ICH, specificity, linearity, range, accuracy, precision, limit of detection, limit of quantitation, and robustness are included. Whereas, according to USP, specificity, linearity, accuracy, precision, limit of detection, limit of quantitation, robustness, ruggedness are included. In this video, we are going to have a look at precision validation parameter in detail.
Distinguish between precision and accuracy. Precision is the closeness of multiple observations to one another. Accuracy is the closeness of a measurement to the true value. Precision, it is the degree of reproducibility. Accuracy, it is the degree of veracity. Precision, a measurement that is precise means that it agrees with other measures of the same thing. Accuracy, if a measurement is accurate, it means that it agrees closely with the accepted standard for that measurement. Precision is independent of accuracy. Accuracy, it is also possible to be accurate without being precise. Precision example, the weight of substance measured is small all the time, so precise. Accuracy example, the average of measurements are close to known value but individually are far from each other.
Precision. The precision of an analytical procedure expresses the closeness of agreement between a series of measurements obtained from multiple sampling of a same homogeneous sample under the prescribed condition. Precision subtype. Repeatability, intermediate precision, reproducibility. Repeatability. It expresses the precision under same operating condition over a short interval of time. It should be assessed using a minimum of 9 determinations covering specific range. Intermediate precision. It expresses variation within laboratories such as different days, different analysts, different equipment, and so forth. Reproducibility. It expresses the precision between laboratories. It is assessed by means of interlaboratory trial defined as ruggedness in USP ISO 17025.
Reproducibility in precision. Reproducibility refers to instances in which the original researcher's data and computer code are used to regenerate the result. It also refers to the result of collective studies among laboratories. Reproducibility, occasionally called between lab reproducibility, express the precision between the measurement result obtained at different laboratories. Reproducibility is not always needed for a single lab validation. However, it is beneficial when an analytical method is standardized or is doing to be used in more than one laboratory. Example, method development in R&D department. Reproducibility should be considered in case the standardization of an analytical procedure, for instance, for inclusion of procedure in pharmacopoeias. Reproducibility is important. It is the only thing that an investigator can guarantee about a study. and the data occurred in reproducibility is not part of marketing authorization. Reproducibility is important because it ensures that the results are correct, but rather because it also ensures transparency and give us confidence in understanding exactly what is the uses. The reproducibility samples are prepared and compared between testing sites. The original researcher's data and computer codes are referred, which it means samples are prepared in the similar manner between two sites and are compared to a previously agrees upon a set acceptance criteria. Recommended data in precisions. There are three recommended data required in precision. First is standard deviation, second is relative standard deviation, and third is confidence interval. Standard deviation, it is a measurement of spread about the mean that is similar to the average deviation. Relative standard deviation, which is also known as percentage relative standard deviation, it is an absolute value of an coefficient of variation, or it is the deviation measurement that tells us how the different numbers in a particular data set are scattered about the mean.
Repeatability and intermediate precision. Repeatability precision is the closeness of the agreement between the results of successive measurement of the same measure when carried out under the same condition of a measurement. In other words, the measurements are taken by a single person or instrument on the same item under the same condition and in a short period of time. A less than perfect retest reliability causes retest variability. Such variability can be caused by, for example, intra-individual variability and intra-observer variability. The measurement may be said to be repeatable when this variation is smaller than a predetermined acceptance criterion. Conditions. The following conditions need to be fulfilled in the establishment of repeatability: the same experimental tool, the same observer, the same measuring instrument, use under the same conditions, the same location, repetition over a short period of time, same objectives. Intermediate precision. The precision obtained within a single laboratory over a longer period of time, generally at least several months, and take into account more changes than repeatability. In particular, different analysts, calibrants, batches of reagents, columns, sprays, needles, etc. These factors are constant within a day, that is, behave systematically within a day time scale, but are not constant over a longer time period, and thus behave as random in the context of intermediate precision. Because more effects are accounted for by the intermediate precision, its value expressed as a standard deviation is larger than a repeatability standard deviation.
Repeatability test procedure. One sample solution containing the target level of an analyte will be prepared. 10 replicants will be made from this sample solution according to the final method procedure. Documentation. Record the retention time, peak area and peak height on the datasheet. Calculate the mean, standard deviation and RSD. Acceptance criteria. The FDA states that the typical RSD should be 1% of drug substances and drug products, 2% of bulk drugs and finished product. HC states that the RSD should be 1% for drug substances and 2% for drug products. For minor components, it could be 5% but may reach 10% at the limit of quantitation. Test procedure. Intermediate precision within laboratory variation will be demonstrated by two analytes using two HPLC system on different days and evaluating the relative percent purity data across the two HPLC system and three concentrations levels, that is 50%, 100%, 150%, that cover the analyte assay method range 80 to 120%. Documentation. Record the relative purity, relative percent, purity percent, percent area and each concentration on the datasheet. Calculate the mean, standard deviation and RSD for the operators and instruments. Acceptance criteria. The assay result obtained by two operators using two instruments on different days should have a statistical data.
Reproducibility can be assessed by using various types of reproducibility tests. First one is analyst versus analyst. By comparing two or more analysts, you can point out inconsistencies in your measurement process. All you need to do is to have more than one analyst to independently perform same measurement process. Next is environment versus environment test, as reproducibility will also most likely be impacted by environmental conditions where the testing and calibration is performed. Therefore, it is necessary to perform environment versus environment reproducibility testing. Method versus method. This type of reproducibility test can be used to determine variance between two or more measurement methods. For example, there may be 4 different methods to perform a particular experiment, but ultimately it should drive to the same result that is expected. Equipment versus equipment test. The reproducibility measurements that include equipmental errors are explained as below. In case of same equipment, the equipment should yield same result in spite of change in model or manufacturer. And in case of different equipments, the equipment should yield expected range of result despite of change of operation procedure. Another common way to test reproducibility of a measurement process is to test variance in measurement results from day to day. This method is great to collect uncertainty in data with only one technician or workbench or both. The only thing you need to change is day or time that test is performed. For example, the experiment is being performed on different days like Monday, Tuesday, etc. or the experiment is being performed on various time slots of a particular day like morning, afternoon, etc. Here is the summary of all the precision parameters. Here is a quick review and comparison of precision and accuracy. As we have seen previously, accuracy is how close a measurement or attempt is to actual or target value, and precision is how consistent our results are regardless of proximity to actual or target value. To exemplify the same, we have 4 dartboard designs as follows. Diagram A represents high accuracy, high precision. Diagram B represents high accuracy, low precision. Diagram C represents low accuracy, high precision, whereas diagram D represents low accuracy, low precision.